Alrighty then, this is muscle fibres. So a lot of the problem here I think is just naming getting the parts right. It's a bit confusing at first what all the bits do, so we'll try and clarify. So if we had a piece of muscle, like say a bicep or something, this is muscle tissue. Now normally we'd think of tissues as being made of lots of cells stuck together. Now it's a bit different for muscles. So rather than it being lots of sort of individual muscle cells that we put together and we say that's a muscle fibre, what these cells do as they develop, imagine that they form kind of one big cell almost. So all the nuclei are still present, but they share one membrane and one lot of cytoplasm. Now each of these individual units, excuse me, it's called a muscle fibre. So let's just redraw that out a little bit better. So if you had lots of these fibres together, like this, I can't really draw loads of them, but we get the idea. That's what a muscle is made up of, lots and lots of these fibres. So it would be multinucleate, but we also have a special nomenclature. So rather than saying that this is a cell membrane around the outside, it is called a sarcolemma. So sarco is the important bit of it. It tells us we're talking about muscles. So the membrane around the outside is called a sarcolemma. Rather than cytoplasm, we have sarcoplasm. And rather than endoplasmic reticulum, or specifically smooth endoplasmic reticulum, we have sarcoplasmic reticulum in here. More on exactly where that is in a second. Okay. The sarcolemma at various points turns inwards. It has these little channels, okay, so there they go inside in 3D, and these little channels are called transverse tubules or T-tubules. Transverse tubules, there we go, or T-tubules. Now why do they have to bend in? Well, they're trying to get into the middle because this is quite a large, it's about 100 microns across. It's relatively large, certainly not on the scale of an individual cell, which might be 5 microns. So anything in the middle here that we need to get to, this sarcolemma needs to be close to it. So it's a way to get into the middle. What exactly is in the middle? Again, let's just draw another myofibril. Another muscle fibre. In the middle are these tubes called myofibrils. And this is basically the actin and the myosin. Okay, all the stuff on the sliding filament theory, which we'll come to in a bit. So these T-tubules need to be able to get right down into the middle, where these to get to these myofibrils right in the centre. Otherwise, the only ones that could get stimulated would be the ones on the outside. So that's what the T-tubule part's for. The process is very similar initially to. I keep drawing these, never mind. The process is initially similar to in axons, when you did neurons, depolarizing and so on. So here's the end of a motor neuron. It releases the vesicles of neurotransmitter, which would then land in receptor sites, and it causes the sarcolemma to depolarize. And it also, the depolarization goes deep down these T-tubules, so it reaches right to the myofibrils. If we pull one of these myofibrils out, and we looked at it, we would see the sarcoplasmic reticulum is in very, very close contact with these things. Very close contact with the myofibrils. As it depolarizes the sarcolemma and down the T-tubule, it causes the sarcoplasmic reticulum to release calcium ions. They're going to be important to us in a second, or in a moment when we come across sliding filament theory. Okay, so that's this structure of the muscle fibrils. So we'll now just have a look at an individual unit of a myofibril, which is called a sarcomere. Now this is the bit with all the lines and stuff. So I'll use a couple of different colours here. Let's use green and blue. So I'm going to simplify this at first. You might not think it's that simple, but it is. I'm going to draw my actin. So the green is going to be actin, and blue is going to be myosin. And the myosin sits between these strands of actin. Now it's not shown on all your diagrams in your book, but down the middle I'm going to draw it this time. The myosin doesn't just float in midair, and this isn't a two-dimensional structure either. If you can imagine, in more of a 3D way, let's put a few more in there. These things are cylindrical, and you can imagine perhaps that being like the actin and the myosin perhaps slipping in between. So it's not flat like we sometimes see it, but that's the way we have to draw it, of course. Okay, what do we need to know to label? These bits at the end are called the Z lines, and the bit in the middle for myosin is an M line. It's not always shown, but it's in there. They're attached to in the middle, like a disc. Now, to remember the various zones, the best thing I can suggest to you is just to remember I can't really call it a phrase, but just remember it like this. I, aha, I. Okay, I, aha, I. And the various zones are as follows. It's easy to work from the outside, so I zone is actually just actin. So in this diagram, it's only that bit. Okay, it's just the actin, that's the I zone. The H one, which sits in the middle, is also easy. It's just the myosin. So it's just that bit is the H zone, okay? And then around the outside, this is why it's easy to work sort of in the way I did it, the A zone is basically where the actin and the myosin cross over each other. So if we contract this sarcomere, if we make it smaller, not really drawn it very well, never mind. You can see now that it's, I've contracted it, it's got a lot smaller than that. Put my myosin in. So the actual filaments themselves, the myosin and the actin, they haven't changed shape, but the sarcomere is contracted. Now when we look at it, we might as well label all of them bits again, Z, Z, and N line down the middle. Now when we get our I, aha, what's happened is that the A zone has got bigger and the H zone has got smaller. Now it's only that it appears to be that because they're sliding past each other and they're crossing over. If you looked at this under a micrograph, it would kind of look like darker bands where the A line is and simply lighter bands where the H zone is. That's kind of what it would look like. Now technically, I've drawn it like this just to show you one of them, but this sarcomere, it doesn't just end there, it carries on, so that what I've done, I could just show you that one sarcomere, but in reality it carries on because each of these myofibrils, the strands, is a string of sarcomeres. Okay, so technically the I zone really could be all of that because it's just actin. So it depends if on the diagram they just give you it like that, or they show you the next one along, in which case all of that is the I band, and it should look a lot lighter on the micrographs.